Welcome back to our study of muscle physiology. So right now we're at the level of the myofibrils, okay? And right here, let me point to what I'm talking about. This structure right here, okay, this is a protein. Maybe you can tell by the ribbon diagram nature of it. And this protein has a special name. It's called, if I scroll over here, that protein that I just showed you was called actin. Okay, and hopefully by now we realize that if you take a bunch of these actin molecules, these proteins, and you string them together like beads on a string, you'll basically get the thin filaments. Okay, and so these proteins right here, these are essentially actin, and this is how I'm going to represent them in this video. All right. So here's what I kind of want to do before we get into the mechanism of troponin and tropomyosin and all that. I want to orient you with these pictures because that's going to be really important. So number one, I just mentioned that these white circles right here, okay, those are representative of actin proteins. And so this is this whole thing right here, okay, this whole thing is going to be basically the thin filament. Okay, it's going to represent the thin filament, and we know, of course, that the monomers of that is basically actin. Okay, this green site right here, and of course, there's going to be many of those. Okay, that's just representative of one myosin binding site on actin. Okay, so these actin proteins are going to have what are referred to as myosin binding sites, and these are the points to which myosin will attach, and we'll get to that fairly soon. So this right here, this structure that I just showed you here on the right, that would be the thin filament, the actin polymer basically, if I didn't have troponin or tropomyosin on it. Okay, what I'm going to do now is I'm going to show you the same picture, okay, but now what I've done is I put all of those proteins on top of it. Okay, now what you should notice first of all is this myosin binding site. Okay, this is essentially going to be covered up by a protein referred to as tropomyosin. Notice the difference here. Notice here I have the binding site, but if I go to the picture on the left where I superimpose everything on it, notice the binding site here is covered up by this orange protein. And this orange protein is referred to as tropomyosin. You may have heard that tropomyosin covers up those myosin binding sites on actin. So literally what it's doing is it's acting as a physical obstruction to the binding site. And so I've represented it here as being not able to see it because it's covered up by the orange tropomyosin. Okay? And the tropomyosin essentially is going to wrap around this protein polymer of actin. And just keep in mind that this kind of extends, in either direction going left and right. Okay, so this is not the whole protein, this is just a segment of it. Okay, now I have tropomyosin, but I also have troponin. So what's troponin? Well, this whole business right here, these three proteins, the red, the yellow, and the blue one, those are all components of troponin. Excuse me, mouth is a little dry. Anyways, I have troponin right here, okay, and there's troponin T, there's troponin I, and troponin C. Okay, these are three units that make up troponin. So let's go over what the troponin T is, the one in red. And notice I've color-coded them. Troponin T is the subunit of troponin that holds tropomyosin. Troponin T is the component of the troponin that holds on to tropomyosin. And the T in the name troponin T corresponds to tropomyosin. So troponin T holds on to tropomyosin. Attached to troponin T, I have this yellow subunit of troponin. That's called troponin I. And I'm not really certain what the designation for I actually is. But basically what troponin I is it physically bonds to actin. Okay, and I tried to designate that here. So troponin I, this yellow one, binds to actin. And then I have this other subunit of troponin, which is troponin C, and the C is going to basically stand for calcium. So in other words, troponin C is the calcium binding protein. Okay, so let's go over that one more time. Tropomyosin is the orange protein that's covering up the myosin binding site on actin. And it's bound by troponin, specifically troponin T, which is the component that binds tropomyosin. Then I have a yellow troponin I, which on the other side holds on to actin. And therefore I also have troponin C, which binds calcium. And this is going to be my setup for formation of the cross bridge. So all this video is going to be devoted to is formation of the cross bridge and how it happens. And honestly, what I'm going to do is I'm going to show you this schematic here, and then I'm also going to show you another method by which we can look at it. Okay? So here's what basically is going to happen. I'm going to have calcium ions. We had already talked about that in the previous video where the sarcoplasmic reticulum resulted in diffusion of calcium out from the lateral sacs into the cytosol of the muscle cell. Okay, and so calcium specifically, remember, is in the 2+ oxidation state. Okay, that's how it exists normally. And you get a massive upsurge of calcium ions in the cytosol when you open the lateral sacs of the sarcoplasmic reticulum. And so here's where the calcium comes in handy. So notice the tropomyosin is on the myosin binding site, okay, on actin. But we don't want that. We'd like it to move away. And so what we're going to do is we're actually going to have this calcium come in. The calcium is actually going to leave the sarcoplasmic reticulum, and it's going to come over here and bind in troponin C, which is the calcium binding component of troponin. Okay? Now, basically, for all intents and purposes, what's going to happen is you're going to go into a high energy state, okay, where you have this calcium that's bound to troponin. That is troponin C. But what's essentially going to happen is when calcium binds to troponin C, it's going to initiate a massive change in conformation of troponin. And so what's going to happen is it's sort of like the foot bone's connected to the ankle bone's connected to the shin bone, connected to the femur, and all that stuff. So when the calcium binds to troponin C changes conformation. Okay? And that's going to translate to a change in conformation of troponin I, which translates to a change in conformation of troponin T. And then when troponin T changes conformation, it's essentially going to move the tropomyosin. And it's going to move the tropomyosin essentially, as you'll see, away from the myosin binding site on actin. So when all of these changes in conformation occur, I end up with something like this. So notice what happened here, okay? Initially, I'm going to have this myosin binding site right here. And as we'll see, myosin is actually going to bind to it. Myosin, I'm indicating by this blue protein. This is supposed to be myosin. In fact, it's the myosin head component of the myosin protein. But the whole point is, notice that I got this change in conformation of troponin C, which translated to troponin I and troponin T, and that ultimately moved the tropomyosin off of the myosin binding site on actin. So hopefully that gives you a little bit of a visualization of what happened. Calcium binds to troponin C, changes in conformation, then move tropomyosin off of the myosin binding sites on actin. And so I end up with this right here. Okay, so I think you can see at this point that myosin right here is going to bind to the myosin binding site on actin. But it will only do so when tropomyosin is out of the binding site. Okay, in other words, when calcium is bound to troponin. But once tropomyosin is effectively moved off of the binding site, remember, that's what happened in the previous step, tropomyosin was moved off of the binding site by troponin, then myosin, this blue protein, is free to bind. Now here's the whole point of this, and what I'm going to do is I'm going to show you the contraction of the myosin head. So basically, imagine this. Imagine that the myosin head, imagine we basically take a javelin and kind of shove it through myosin like that. Okay, so you're looking at basically the tail of the javelin. So it's basically going into your screen right there at the red dot. And let's imagine that myosin is basically able to rotate about that point. Okay, so hopefully that makes sense. And so what myosin will actually do in the presence of adenosine diphosphate, and we'll talk more about that later, is the myosin head will actually rotate like this. Okay, so essentially it's going to rotate about that point to where now you rotate it up like this, right? So hopefully you see the difference, and I've tried to denote this with these gray lines. So let me zoom out at this point so hopefully you can see what happened. Okay, so notice if I'm looking at this point right there, okay, that's the same point, notice that the myosin head actually rotated a little bit. It kind of changed conformations and moved its top part towards the left. But notice it's still bound to actin, so it takes the actin with it. Okay, and hopefully you see that. And so what happened is notice the whole protein moves, in this case, it's towards the left. Okay, and we'll talk about the implications of that in a minute. And notice what else happened is when this whole protein moves towards the left, this whole actin thin filament moves towards the left, essentially what you get is a 10 nanometer movement. Okay, so not only is it this side right here that's moving to the left, but I also have this side up here that's moving to the left. And hopefully you see that, once again, it's about a 10 nanometer movement towards the left. And essentially what's going to happen is the power stroke is going to occur. So that's a good piece of terminology. So this movement right here, okay, let me write this down. This movement where the myosin head rotates and pulls the thin filament with it, that's called the power stroke. That is called the power stroke. And the power stroke essentially is where the myosin head kind of rotates a little bit, and hopefully you can see that here. The myosin head is rotating about some point, basically, for all intensive purposes, and it takes the thin filament with it. And so in this case, overall, the thin filament moves towards the left. Now, if we're pulling the thin filament in that direction, essentially what we're doing is we're pulling the thin filament towards the M line. That's going to result in an overall contraction or shortening of the muscle fiber. But we'll look more on that in a few videos. But hopefully this made a little bit of sense. And now let's look at it from another perspective. So I got this picture off of Google, and I thought it was pretty good. So hopefully you can kind of see this in two ways. Okay, so this right here, this is sort of at rest. This is the situation that we have at rest, and this is basically during contraction. Okay, and I think you'll see what is going to happen here. Okay, I think I have the wrong brush. Okay, there. Okay, so here's what's going to happen is, notice that tropomyosin, the purple protein in this case, is on these binding sites for myosin on actin. And these binding sites, I'll kind of label them, and hopefully you can see what I'm talking about. They're these things right here. It'll work. These things right there. And hopefully you see those things. And notice tropomyosin is on those binding sites. And also notice that troponin, specifically troponin T, is holding on to tropomyosin for dear life. But notice, once calcium binds to troponin, notice how this tropomyosin has sort of rotated off of the myosin binding sites on actin. And so now what you have, and I'll go ahead and circle some of these, is you now have exposed myosin binding sites on actin. In fact, right here, these two binding sites have now been bound by myosin. And so from there, what's going to happen is the myosin is going to rotate, and it's going to move the thin filament towards the M line. And we'll talk about that in another video. But hopefully, this gave you a little bit of intuition on what's happening. So let's do a quick recap of this process. So number one, I have tropomyosin, which is on the myosin binding sites on actin. Notice, oops, that was black. Let me get orange. So notice the tropomyosin is on the myosin binding site on actin. Notice here I don't have the tropomyosin on there because I haven't drawn it yet. But here I have the tropomyosin. It's on the myosin binding site. Okay, and troponin T is holding it there. That's the one in red. Troponin T is also attached to troponin I, which is physically attached to the actin, thus stabilizing it. And then I have troponin C in blue, which is the calcium binding component. So basically, you could sort of imagine as step one would be calcium binding to troponin C. And what that's going to do essentially is it's going to cause changes in conformation in troponin, which effectively, step two, it moves tropomyosin off of that binding site. And so then you're now free to bind myosin. That's step three. You're free to bind myosin to the myosin binding sites on actin. And that's because this guy right here, the tropomyosin, is off of the myosin binding sites. And that's essentially what we saw over here in this picture. Once we got tropomyosin off of the binding sites, immediately myosin binds. So hopefully that makes a little bit of sense. And then what happens is something referred to as the power stroke. And so notice myosin essentially rotates, and it pulls the entire thin filament with it, because it's attached to the thin filament through actin. And so notice what I get essentially is approximately a 10 nanometer migration towards the M line. Okay, and that's per power stroke basically. Okay, so hopefully that video made a little bit of sense. In the next video, we're going to do some stuff with myosin and the myosin cycle. See you in the next video.